Have you ever stopped to think about how your body knows how to function at every moment, or what tiny biological mechanisms make you you? Imagine if every cell in your body had an instruction manual telling exactly how to grow, function, and repair itself. Well, this manual does exist, and it's called DNA. DNA is made up of four chemical letters, A, T, C, and G. The specific order of these letters form specific genes, which give the cell instructions such as how to divide or make proteins. When the sequence and instructions are correct, the cell functions normally. But if even one letter is missing or misplaced, the entire meaning of a gene can change and give the cell incorrect instructions. This can lead to genetic diseases such as sickle cell anemia, cystic fibrosis, or certain cancers. For a long time, scientists were really good at reading DNA, but not at changing it. Early gene editing techniques were slow, expensive, and messy, kind of like trying to fix a typo in a book by ripping out entire pages rather than fixing that one letter. What scientists wanted was a way to target a single location in a genome to edit a specific gene's instructions. That's where CRISPR comes in. CRISPR is based on a natural defense mechanism found in bacteria. Bacteria are constantly being attacked by viruses, and CRISPR acts as their immune system. When a bacterium is infected by a virus, it saves a small portion of their DNA, so that way when the virus attacks again, it can easily recognize the invader due to that saved sequence, and it can quickly cut its DNA apart. Scientists realized they could reprogram the system to cut out any DNA sequence they want, not just viral DNA. To do that, CRISPR relies on two main parts. The first is something called guide RNA, which scientists design and modify to match a specific DNA sequence. Think of it as a DNA GPS because it leads CRISPR directly to the part of the genome that needs to be cut. The second part is a protein called Cas9, which acts as molecular scissors to make that cut on both strands of the DNA once the guide RNA has matched its sequence. Now, the important part is that once the DNA is cut, the cell will rush to repair it. This repair process is what makes gene editing possible. Sometimes the repair will introduce small errors that can turn the gene off, but scientists can implement a corrected piece of DNA called a DNA template strand. The repair process will copy the strand, and according to what the scientists want, it can repair a mutation or even implement new genetic information. Because CRISPR is fast, relatively inexpensive, and extremely precise, it has transformed biology. Researchers are now using it to study genes, develop disease-resistant crops, and explore treatments for genetic disorders, including ways to edit immune cells that can better recognize and attack cancer. However, this comes with serious ethical questions. Editing cells within an individual affects only that person, but if we decide to start editing embryos, that could pass changes on to future generations. This raises concerns about safety, consent, and where to draw the line between treating disease and enhancing human traits.